Hope everyone's having a great day and has had a great meeting so far. I think I know most of you. I'm Michael Warner. I'm the co-founder and executive director of the Alliance, and it's great to have all of you. I'm particularly pleased to introduce our next speaker, someone that I've had the pleasure to work with over the years, and on a personal level, really a wonderful colleague. Celia Witten is the official title, is long. It's the director of the Office of Cellular, Tissue, and Gene Therapy Center from the Center for Biologics Evaluation and Research at the U.S. Food and Drug Administration. You'll usually referred to as OCTGT in the Center for Biologics. And prior to that position, she's held a series of posts at FDA. She was also a practicing physician at the National Rehabilitation Hospital in Washington, D.C. She's board certified in physical medicine and rehabilitation. I really need to mention that when ARM first came into being several years ago, one of the first things we did was bring a delegation to talk to the leadership of the FDA. We met with the commissioner, Dr. Hamburg, and senior staff, and we also, of course, met with Dr. Witten and her senior team, and that really was the beginning of a wonderful, productive, and collaborative relationship. I'm very pleased to say that the Alliance has had a very good working relationship with the FDA. Dr. Witten has always been very welcoming of us, interested in listening to the challenges that our members, all of you, face, and trying to work on working together with us on some practical solutions, as well as we've had some initiatives about educating companies and sponsors about sort of the best ways to bring an application together. So we're very grateful to all of the support we've gotten from Dr. Witten. She's going to talk to us this morning, and then we're going to have a regulatory panel. So we're really very much looking forward to the discussion over the next hour or so. So without any further introduction, I'd like to call up my colleague, friend, and our special speaker this morning, Dr. Celia Witten.
Thank you for that introduction, and also for inviting me to come and speak today. I'm going to give a brief overview of the office, and because I know not everyone is familiar with exactly how we're structured. And then I've picked some topics in product development, regulatory policy and guidance, and a few special programs to touch on. So I'm not going to try to cover a regulatory framework from A to Z, but some topics of interest. So just to mention, our office, which is highlighted in yellow, is one of the 3 offices that do what's called product review in the Center for Biologics. And we work with our colleagues in the Center for Drugs and the Center for Devices, because obviously there's a lot of cross-cutting issues in this field. In our office, in addition to regulatory review, we work on policy and guidance development. We have an active program for international harmonization or convergence activities, and also standards. We do a fair amount of outreach, and we also have some publications and mission-relevant research, which I'm going to talk about a little bit. So these are some of the products in our office that we have oversight responsibility for. Now, I've called it products here, but a lot of these are investigational, obviously. And these categories aren't meant to be, they are overlap in these categories. Obviously, gene therapies, in which I know there's a lot of interest, are also, many of them are cell therapies as well. But this gives you the flavor of what we see: stem cells, somatic cell therapy, gene therapy, therapeutic vaccines for cancer, and some devices and combination products. I feel like I'm echoing. Can you hear me clearly? Yes? Okay. Okay, and so people always want to know about, how many products we see or how many things under investigation we have. So this graph shows you the blue is the total, and it's broken down by cell, gene, and other. And you can see that over the past couple of years, there's been an increase in the office and how many new INDs submissions we've seen. And I think this really goes along with the excitement in the field and what some of the other speakers were talking about in terms of industry investment. So we're really excited about the products in our office, and it's clear that the community also shares that excitement. I think that's reflected in an increase in other areas as well. So specifically about product development, I'm going to talk about two topics really briefly. One is product characterization, the MSC consortium, and the other is a couple of what we see as advances and challenges in the gene therapy area. So for all biologics, including cell and gene therapy, regulatory considerations include safety, efficacy, purity, and potency. I'm sure you all have heard those terms: oversight of the product and the process, quality control, and reproducibility. But this isn't meant to be a regulatory primer, so I'll just say that if you're a sponsor and you want to bring a product for study, for example, what would be your best course of action? And this is the only slide I'm going to have on what you should do if you actually want to, come and talk to us about your IND, which is first do a little homework, find out what guidance there might be in some specific investigational areas, both in terms of the clinical area and also general cross-cutting guidances. See if there's any standards that might be useful, and have a pre-submission meeting with the appropriate work unit at FDA. And we have a very active pre-IND program, which the number of meetings we've had, if I showed that slide, the numbers have gone way up in harmony with the numbers of IND submissions that I showed you on the other slide. So I want to talk specifically about a product characterization topic, and this isn't meant as a discussion on MSCs. It's meant as a discussion on the importance of product characterization. So I'll just say that we hear that, MSCs is an area of interest, and we hear a lot, MSCs are known to be safe. We know this about MSCs, we know that. And so we decided to look at what people mean when they talk about MSCs, because I think it's one of those areas where product characterization is important. So this, I think, is a very good effort on the part of ISCT, the International Society of Cell Therapy, they proposed an industry standard to harmonize MSC product characterization. This is a couple years back, and I think it, it's a good place to start, and it was our starting point for the two research projects that I'm going to talk about. So what they did was they said these are the markers you should look at, the criteria you should look at to identify an MSC. The first part of the research I'm going to talk about briefly is a lab consortium, and so I mentioned that we have labs in our office. A number of researchers got together and looked at MSC cells, the same cells in different labs in our program, and looked at different parameters to characterize the cells to see what they could learn about them and how that related to the characterization tool that ISCT proposed. So these are from different donors and different passages, and you can see this is just I'm just showing a couple example parameters that they measured. So what they did was they took a number of cells from different donors and looked at them at different passages to see how similar, or whether there were parameters that could differentiate the cells. So here's one that shows cell size. This one looks at colony-forming units, and you can see again a difference between donors and also for a given donor between passages. Here they looked at adipogenic activity and also saw differences between donors and between passage sizes. So then they applied the consensus MSC markers from the ISCT standard. That's what I showed the first slide on. And you can see that although there were differences seen on the markers that the research group studied, the consensus MSC markers were not able to distinguish those differences. So that shows that there are some differences between these cells that aren't delineated by the markers that were the consensus MSC markers. So what about our INDs? We also took a look back at the product characterization INDs, and I want to just give credit to Mike Mendocino, who's the commissioner's fellow referred to in this slide. He was a fellow at FDA. He's now working as a reg affairs. He's left FDA, but he's here. So if you have any specific questions on this article, you can ask him. Anyway, I should say he's working for Mesoblast now. So he reviewed, together with the team in our office, product characterization submitted in MSC-based INDs from 2006 to 2012. And this is published. I have the reference citation here. So I'm really only going to give an overview of some of what was looked at, but the basic question was what cell surface markers were used in our INDs, and they were looked at these various stages, product development, during manufacturing, for final product release. And so here are the top 20 cell surface markers used to characterize MSCs in our INDs. And on the right of this slide are the ISCT criteria that indicates the range in ISCT and the consensus markers that they identified. So what you can see is not all of the INDs use the ISCT consensus criteria to characterize their cells. That's not to say it's good or bad. It's just to say that they're not, they weren't considered, by individual sponsors who made selections didn't select those. And they didn't all select the same one. There's a lot of heterogeneity, and not all of them described a range, and the ranges were different. So this just makes the point that when people ask us to go back and do data mining of our INDs, the products may or may not be similar. You really can't tell from what we have submitted. There was also manufacturing diversity in the INDs in terms of whether fetal bovine serum was used in the media, growth factor was added, atmospheric oxygen during cell culture, and these all could have affect the biological characteristics of the MSCs. So these are challenges for MSC clinical translation, and the conclusion is they're diverse in source, characterization, and manufacturing. So when we do reviews of these, we review each one based on its own merits. There's not a current requirement for consistent nomenclature, but it's something that the industry might think about, maybe not with this in this area, but it's a challenge in general to look across studies or across scientific publications for that matter and be able to really compare one to the other. And this is just a slide of some of the publications from this group. So now I'm going to move on and talk about gene therapies. This pie chart just tells you the diversity of vectors or types of gene therapies we've seen in the office recently. This is a current slide. So there's a number of challenges in this area: better cell engraftment, more efficient. Some are in clinical and some are in the manufacturing end, like more efficient vector transduction and need for vectors with improved safety profile. And then certainly clinical trial methodology is always a challenge for rare diseases. However, with all those challenges, I will say that there's a lot of excitement in the gene therapy area, and we've heard about some of it at this meeting. But there's been, from our point of view, there's been a number of vector and product improvements, use of preconditioning regimens for increased cell engraftment, and there's been some promising early phase results in a variety of indications. So moving on to regulatory policy and guidance development. These are some recent guidances. The preclinical assessment of investigational cell and gene therapy products was finalized. We have a draft guidance for industry on considerations for the design of early phase clinical trials of cell and gene therapy products. We took that draft guidance to an advisory committee for comments as well as in the open public session, and we're in the process of finalizing it now. There's a draft guidance on design and analysis of shedding studies for virus or bacteria-based gene therapy and oncolytic products. that was issued this past summer. We are taking that one to an advisory committee in November for comments, and obviously there's an open public comment period as well. And that is for the question of, well, what it says, design and analysis of shedding studies. And then there's the environmental assessment draft guidance, which was issued this past summer. It's not going to an advisory committee, but we will be looking at the comments and finalizing it. And we have a guidance agenda that gets published every 6 months that tells you what our priorities are for guidance development in the ensuing 6 months. So this is what was published, I think, last June, and we're hopefully, we're certainly working hard on these. So whether we'll meet the target of December or whether we'll miss it by a couple of months, I can't say. But these are on their way. One is on minimal manipulation for tissues. One is on the same surgical procedure exception, which is one of the exceptions in our regulations. It tells you if something's done in the same surgical procedure, it doesn't meet the criteria of being human tissue. And the other is specifically on adipose tissue. So these are all things we've gotten a lot of questions about. And usually we set the guidance agenda based on what we know industry's been asking about and what we need to provide some more clarity around. So I mentioned the first advisory committee that is happening this November on design and analysis of shedding studies, and we're also going to be presenting the Dear Gene Therapy IND, or Master File Sponsor Letter, that's been part of the INDs for gene therapy for quite a while. And I want to mention that our last advisory committee, the other one we had this year, was on 2 topics. One I mentioned, which was the early phase clinical trials, and the other was on oocyte modification and assisted reproduction for prevention of transmission of mitochondrial disease or treatment of infertility. I don't think there's time to go into a lot of detail here. For those of you who already are familiar with this meeting, though, you might be interested in knowing that as an update to the number of questions we got about the social and ethical issues, that we have commissioned a National Academy of Science consensus report, and you can read on their website a description of their work in that area. So that is an ongoing project. We have a number of international activities working on either harmonization or convergence activity, and there's not really enough time for me to talk about each of those individually because I want to leave some time for questions. But we have a regular dialogue called a cluster meeting between us, EMA, and Health Canada, and that is, I think we meet every 2 months, and we talk about both general topics and specific matters. We also have had a number of our sponsors have requested parallel scientific advice from FDA and EMA. That's not specifically a procedure for regenerative medicine. It's something FDA and EMA established, but a number of our sponsors have taken advantage of this route. And then there's the International Pharmaceutical Regulators Forum, which happens sort of around the margins of ICH. And these are more convergence. The groups, the regulators that are involved in that forum span groups with a range of maturity of regulatory frameworks. And the goal of those discussions is really, to figure out what the frameworks are, to have some discussions about some basic things like terminology, and try to reach a common understanding of what some of the issues are. So the last topic that I want to cover briefly are the expedited programs and expanded access, because there is always a lot of confusion and conflation of these terminologies and what those programs are and what they mean. So I'm just going to briefly talk about them and mention what they are and, what they're for. So FDASIA, the most recent reauthorization of PDUFA, was signed into law a couple years ago, and it had a couple of sections. One was enhancement of accelerated patient access to new therapies, which clarified some provisions and expanded the use and scope and evidence FDA can rely on. and breakthrough therapies, new designation with goal of expediting development of drugs that offer substantial improvement over existing therapies. And that's the one that we get a lot of questions on, so I'm going to have a little slide on that. But first I want to just say we have had in place already fast track, accelerated approval, and priority review. So what those are, the fast track is designed to facilitate development and expedite review of drugs to treat serious diseases and the potential to fill an unmet medical need. And that it doesn't change the type of evidence. It's just something about, the timelines and so forth. Then there's accelerated approval, and that's where you can get accelerated approval based on a surrogate endpoint under certain conditions, and you need to do a confirmatory trial. So that one actually speaks to the type of evidence, not that there doesn't have to be safety and effectiveness evidence, but what you can look at as an endpoint. And FDA has certainly over the years made use of accelerated approval. And then priority review speaks to the time frame. So there's a standard review time frame and a 6-month review time frame if you get priority review. So that's the criteria listed here. So the accelerated has to do with the endpoint. The others aren't so much with the evidence, but just with the process. So breakthrough therapies, first I have the definition listed here. So those are for those are for products that are intended to treat a serious or life-threatening disease or condition. And preliminary clinical evidence indicates the drug may demonstrate substantial improvement over existing therapies on one or more clinically significant endpoints. So we've had a number of applications in this area in our office, and FDA has as a whole. But speaking to our office's experience, the ones we've granted have been where there's been some actual clinical evidence, first of all, some actual clinical evidence in the product. So we've had submissions where the sponsors say we are going to make this product in this way, and when we do, we expect it to work like this. That doesn't count as preliminary clinical evidence, so those we haven't granted. And then there's a number of them where I can understand why FDA is always confusing. And so I can understand why people are submitting these. I'm just saying we're not granting them. And also the preliminary clinical evidence and the substantial improvement, we've been interpreting this really that when you look at it, either it's a phase 1 randomized trial, small trial that at least gives you an idea of the effect size, that you have something to go on, which is a substantial improvement, or there's something which is so different or striking in terms of its effect that that it would be substantial improvement. I mean, just something that you just would not expect or see in nature. So the ones that have been problematic have been things like, aside from the product hasn't been made yet, if someone is looking at time to event and they do a single-arm trial, I think those are very hard to interpret. So a lot of the ones that we have not granted, it's been because it's been a time to event single-arm trial. So either a small randomized trial or something that's really, it just really different in terms of its improvement. Those are the kinds of things we've granted. So expanded access, I'm just mentioning this. This has been around for a while. This isn't new. And there's a guidance on expanded access, and I think it's either that guidance or there's a separate guidance that speaks to requests for reimbursement and expanded access also. So this is for use of an investigational drug outside of a clinical trial, basically for treatment. And so if the sponsor has completed their clinical study, and while they're waiting for the results to come in, that puts them in one scenario. They might get a treatment IND. Sometimes there's groups of patients that don't fit the criteria for the clinical trial that's ongoing. We encourage the sponsors, if there's a group who might be not eligible for that trial, but might be good candidates for the product, to actually, consider doing another study. But sometimes we'll end up granting an intermediate-sized group access trial, and certainly individual trials also. We don't want to keep people from getting something, but on the other hand, we want to make sure that there's a development program for the product. So that's always a challenge for sponsors as well, I believe. So we have, for those of you who want to know in detail about our regulatory process, we have a very nice but rather extensive webinar series, which covers a lot of our, our policies. And I have here the source for regulatory questions and just general useful websites and my contact information. And I'd be happy if the organization wants to share my slides. If anyone requests them, that would be fine. They could be posted. So if there's anybody who wants it, you can see if the meeting conference wants to post them, that would be totally fine. So that is all I had planned to say, and that leaves about 6 minutes and 7 seconds for questions. So are there, I've been watching the time. So are there?
Anybody have questions for Dr. Witten? Come on, we know you do.
From Stanford. I had a question. I think a lot of people who are using cell-based therapies might have this, and it has to do with the inclusion of serum and maybe the FDA's stance on the inclusion of bovine serum albumin or just serum in general in the culture of cells. I know that you had it on one of your slides, and so I'm wondering if you have any general guidance you can provide in that area.
Well, what I would say generally, specifically, you probably want to talk to the reviewing product office and ask them your questions specifically. But just in general, I would point you to the, I think it's the Xeno guidance. I mean, we have guidance that speaks to some safety issues in that area. It talks about knowing about the source and that kind of thing. But probably you're best off contacting Lori Toll is one of these numbers here, and she can set up a phone discussion about your specific question with the reviewers.
Hello. Just a quick question on the review process. Once you have an IND approved at the FDA, and you need to go through, our product do change through the manufacturing.
I'm sorry, I'm having trouble. Say that again. Once you have an IND.
Once we have an IND approved, and our products usually do change a little bit through a development cycle, and then you need to go through sometimes an amendment of that IND. There is no predefined timelines, as I understand, for the review of those amendments, which makes the conduct of a clinical trial complex to plan for. Is this something that the FDA intends to address one way or another by having some more predefined timelines for the review of those amendments?
Well, generally, so when amendments come in and they're not, the reviewers don't have a heads up, they need to look at this for this reason, they may take a while. But I would suggest calling the product reviewers or the regulatory project manager and explaining what it is you're sending in and why you need it reviewed, what your need is. And then you'll get a more helpful response probably than just sending it in. I mean, because that's what happens. Sometimes people just send things in and it's in a queue, and it's just not recognized because it hasn't maybe been looked at, that there's some time sensitivity about it. So I would suggest calling the regulatory project manager and explaining what your needs are. Because they will help. We do deal with that issue.
Thank you.
Hi, I'm from Celadon. We recently received breakthrough therapy. And so we're wondering what kind of changes we can expect from the regulatory process that we've been working with you all on.
Oh, yes. Thank you for asking that, because that's actually what I meant to say. Because I had talked about what the other, I appreciate you asking that question, because I talked about what the other designations meant or got people. And basically, the breakthrough therapy does not change it doesn't change the evidence level for the product. What it changes is it changes the access to meetings and advice. And so, I mean, I think our office is fairly, interactive anyway in terms of meeting with people, but, we do sometimes, if we've had a couple, there are criteria, and really sponsors should get one pre-IND meeting, for example. That's it, one meeting like this. But if it's breakthrough, they have a breakthrough designation, then they get more access for meetings and, more questions. Sort of earlier, we might consider bringing in other offices if we need to earlier in the development process rather than waiting till the end. So if there's some needs that a sponsor with breakthrough has that they've identified that they think they need advice on and they're not getting met, they should be able to get them met. So that's basically the difference. It's access to, to more advice and meetings. So that because one thing we don't want to have happen, for example, is that someone gets breakthrough and let's say, just for example, their clinical development program is proceeding at one rate, their product development program is lagging because they're doing so well, they're recruiting their patients and everything. So we want to make sure that doesn't happen. And so definitely, more interaction would be helpful for those sponsors.
Last question.
Okay. I'm Steve Davis. I'm with STEM Biosys. A lot of us talk about minimal manipulation, and I'm wondering if there are any changes on the horizon or how you view this, because that turns out to be a big challenge for many of us who are trying to expand cell lines, thinking that now we're going to be, it's going to be a drug, and you go through the different phases of clinical development for that. Is there any thinking that would tend to modify what seems to be the current position, or that going forward would seem to put cell therapies in a slightly different category?
No, I don't think you can anticipate a change. The guidance, I think, will clarify what we think is already existing policy.
Okay, please join me in thanking Dr. Witten.